Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1963 witnessed the significant contribution to the burgeoning field of biophysics, a contribution that challenged prevailing models of enzyme, kinetics and laid the groundwork for a deeper understanding of complex, biological systems. This pivotal work, authored by Manfred Eigen in that year, bore the title Impossibility of a Simple Kinetic Description of the Evolution of a Complex Enzyme. Eigen, a German biophysicist who would later be awarded the Nobel Prize in Chemistry in 1967, meticulously dismantled the prevailing view that enzyme evolution could be adequately described with simplistic kinetic models. His paper, a harbinger of his later explorations into self-replication and information theory in biology, forced a reconsideration of the assumptions underpinning the study of enzyme behavior. Before Eigen's seminal paper, the dominant paradigm in enzyme kinetics rested upon the principles established in the early 20th century by Leonard Michaelis and Maud Menton, and later refined by Gwendoline Briggs and J.B. Haldane. These kinetic models, while providing a valuable framework, operated with a degree of simplification that, Eigen argued, was increasingly inadequate in the face of growing biological complexity. The Michaelis-Menton equation, for instance, posited a straightforward relationship between enzyme, substrate, and product, often assuming a single, rate-limiting step. However, as biochemists continued to explore the intricate world of enzymes, particularly those associated with multi-step processes like the synthesis of larger biomolecules or regulatory pathways, the limitations became increasingly clear. These enzymes, far from exhibiting singular catalytic actions, often manifested intricate allosteric properties, exhibiting cooperativity. The idea of a static, predictable enzyme structure became obsolete. A more dynamic model was required. Eigen's work began in this intellectual milieu. The essence of Eigen's argument resided in the inherent complexity of enzyme evolution and the inadequacy of simple models to capture it. He posited that enzymes, particularly complex ones, had evolved through a multi-stage process, subject to the pressures of mutation, selection and environmental adaptation. This evolutionary process, he contended, created a landscape where simple kinetic descriptions were simply incapable of capturing their underlying dynamics. The paper meticulously detailed and formalized these arguments. Eigen introduced the concept of a selection value to quantitatively describe the advantage gained by a particular enzyme variant, showing how this selection value changes over time and how this change depends on a variety of internal and external factors. He argued that the simultaneous action of multiple reaction steps rendered the application of simple differential equations to describe enzyme behavior a deceptive oversimplification. The paper explored the challenges of modeling such systems, particularly the difficulties in accurately determining rate constants within the complex multi-step pathways. Furthermore, Eigen highlighted the sensitivity of enzyme kinetics to slight variations in the conditions, which would necessitate consideration of the distribution of enzyme structure as opposed to a single, static conformation. The immediate impact of Eigen's work was a profound shift in the perspective on enzyme kinetics. It spurred a move away from reliance on simple models towards more comprehensive and nuanced approaches. Prior to Eigen's contribution, the focus of research frequently aimed at reducing the complexities of enzymatic reactions to manageable representations. The success of the Michaelis-Menton model, in particular, served as a kind of template for understanding enzyme behavior. Eigen's work, however, demanded that researchers embrace the intrinsic complexity of enzymes as characteristic. This pushed the development of kinetic models that incorporated allosteric regulation, cooperativity, and the impact of the enzyme structure on its function. Scientists started to think about enzyme landscapes. They started to consider the importance of enzyme conformation and how variations in conformation impacted enzyme behavior, and how that behavior could be modeled. This changed the research agenda. The emphasis shifted to methods of quantifying the kinetics of complex enzyme systems. The implications of impossibility of a simple kinetic description of the evolution of a complex enzyme reverberated through many fields. Eigen's formalization of the interplay between mutation, selection, and environment provided the conceptual basis for understanding the evolution of biological molecules. For example, this impacted the study of protein folding, as it forced researchers to recognize the vastness of the possible conformational space for a protein and to consider the role that evolution played in selecting specific, functional conformations. The work also influenced the study of metabolic pathways. Researchers had to grapple with the complex network of reactions, enzymes, and regulatory mechanisms that operate within living cells. This fostered the development of sophisticated computational models that could simulate complex biochemical systems. Eigen's work was also linked to the broader field of information theory, which would become a central part of his subsequent research. He recognized that the information content of a biological molecule was crucial to its function, and that this information could be subject to evolutionary pressures. He extended his ideas on enzyme evolution to models of self-replication. Eigen's collaboration with Peter Shustad, specifically the concept of the quasi-species, built directly upon the foundations laid in the 1963 paper. The quasi-species model, which describes the evolution of populations of replicating molecules, incorporates the principles of mutation and selection. It demonstrated the influence of error rate on the stability of genetic information. It offered a mathematical framework for understanding the origin of life and the evolution of viruses, representing a culmination of the ideas presented in his 1963 paper concerning the limits of simple kinetic descriptions. The reception of Eigen's work was initially subtle, though its influence has become profound. The concepts presented, at first, did provoke a rethinking of the accepted methods. The scientific community, accustomed to the more tractable simplicity of the Michaelis-Menten kinetics, found Eigen's arguments, at least in the beginning, somewhat difficult to immediately integrate. The limitations of the simple model became clear slowly. As techniques improved to probe the structural intricacies of enzymes, the framework Eigen presented became more and more important. Some researchers began using more sophisticated computational models to account for the multiple steps and allosteric interactions in the evolution of complex enzymes. Eigen's work also faced challenges. For example, critics questioned whether the concepts were overly abstract, without immediately obvious experimental validation. However, as experimental methods improved to explore the dynamic nature of enzyme behavior, the ideas contained within the paper received ample support. Eigen's 1963 paper was clearly influenced by several preceding works. Certainly, the work of Michaelis, Menten, Briggs, and Haldane formed the foundation upon which Eigen constructed his arguments. Their kinetic models, representing a significant advance in the study of enzymes, provided the very framework that Eigen subsequently challenged. The development of physical chemistry and thermodynamics, which helped establish the principles governing chemical reactions, provided the conceptual tools and language employed throughout Eigen's work. The increasing availability of powerful spectroscopic techniques and the advent of new methods for characterizing enzyme structure also enabled a level of analysis that was far less accessible to earlier generations of scientists. Eigen was one of the first scientists to fully appreciate the impact that these advances would have on the study of enzyme behavior and the limitations of assuming static structural representations. The profound influence of Eigen's work is undeniable. It spurred numerous subsequent contributions across diverse fields. His ideas on enzyme evolution directly fertilized the development of computational biology. The emergence of bioinformatics, with its methods for analyzing large datasets of genomic and proteomic information, can be directly traced to the recognition that enzyme function could be described at multiple levels of complexity. The development of drug design platforms, for instance, makes extensive use of computational methods that simulate enzyme-substrate interactions and predict the effects of mutations. The understanding of enzyme evolution has also helped in the development of new technologies, like enzyme engineering, which utilizes directed evolution to improve enzyme behavior for specific applications. Eigen's 1963 paper represents a pivotal moment in our understanding of enzymology and the evolution of biological systems. Through meticulous analysis, he demonstrated the inadequacy of simple kinetic models for accurately capturing the complex dynamics of evolving enzymes. His work instigated a paradigm shift, propelling scientists to embrace the intrinsic complexity of biological processes. The paper's legacy is enduring and evident across a wide range of scientific fields, from biophysics and biochemistry to computational, biology and evolutionary theory. Eigen's contribution underscores the crucial importance of acknowledging the multifaceted nature of biological phenomena, and its impact continues to drive progress in our understanding of the molecular mechanisms that underpin life. It remains a testament to the power of theoretical insight to transform the scientific landscape and to open up new avenues of investigation.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.